We live in very different worlds. Genomic medicine, of course, infers that we're going to bring together genome stuff and genetics and clinical practice. And I feel like much of the time we know so little about each other's worlds, even though we're here probably nestled together more closely and more intimately than in any other institution in the world. If you know, nowhere else can match our sheer mass of individuals on both sides of that fence. So I think we have this extraordinary opportunity and just enormous barriers to what we do. So that's why I jumped at the chance to come and talk here. And I feel like every time we have a dialogue across this fence, or every time we do something, a collaboration, we make one more little dot in the big mosaic that we're going to build. And maybe one day we'll all stand back and look at that mosaic, and then we'll know exactly what genomic medicine is. In the meantime, I think we kind of have to struggle through to make our own definitions. So what I really want to do today is to give a bit of a reality, some reality to some of the elements of the whole enterprise, and set the framework from the genomic point of view, and also from how the study of genetics is unfolding in informing human disease. And so, of course, it's all directed to this magic model where somebody walks into your clinic, and I guess this is the near-term model where you send their sample over for a complete human genome sequence, and you use that to inform your decisions. The further-term model will be that they'll actually walk in with that little device, or you'll look at it online, and they'll already have logged in their family genetic information, their own DNA sequence, and you'll just have to look it up to get it. If you subscribe to that model, which of course we all did at the beginning of the Human Genome Project, when there was a lot of hype, we offered a lot of publicity. You had to subscribe then at that time to this pretty simplistic model of how all this was going to work. That is that there would be discovery. I mean, there would be a primary data discovery, a sequence of the human genome. This would easily lead to discovering genes and inferring their function, and we would just use that information there to modulate and cure disease and do diagnostics and prognostics. Of course, we all knew that was a very naive model, and that had much more complexity to it, and that in fact this big box on the left actually represents all of biomedical research and what everybody's been doing all along. And I think what we've been doing since is really adding layers and layers and smaller text to this diagram, acknowledging the importance of integration of this simple genomic, the naivete and the directness of genomic approaches to things with the complexity of biomedical research. Within that, the technology has been critical. That is that every step in the development of that whole model has been enabled constantly by technology, and that is continuing to be the theme. And at each point, the only real reality check we've had is to sit back and look and say, well, what have we achieved in terms of development of data sets and development of knowledge about individual disease? So that's the framework, as I already said, that I want to address today and put it in the context of these three questions. How are we doing with this idea that a lot of DNA sequence and analysis of genomes and the follow-up from that can actually drive knowledge discovery? And in particular, how are we doing that in the context of genetics and disease? And gee, what should we do next, particularly here at Baylor? So the first question is actually pretty easy to answer. We're really doing pretty well. This is my favourite slide from 20 years ago, when we did a first PCR direct analysis of a gene and found mutations at the heterozygosity state that caused a direct disease. And so this was like a nice illustration of the first application of polymerase chain reaction and fluorescence sequencing to find all the mutations, and to say that the rest of the gene wasn't affected because we looked at every base in it, if indeed the rest of the gene wasn't affected. So we've gone from that now through sequencing the whole human genome, and here's the scene from the White House where everyone had a group hug saying, it's all done, and the private and public sectors have all agreed to disagree about certain things, but the data speak for themselves. And then that's the shortest history of the Genome Project I've ever been able to deliver. It's coming down to about 10 seconds. And of course, after that, we came home and figured that we didn't, we had a lot of TCs, A's and G's and didn't really understand them, and asked ourselves the critical question of, how could we begin to interpret these data and eke out what was inside of them? And pretty much came to the conclusion that what we needed more than anything else was actually more sequence data in order to drive comparative genomics and to figure out some sources of genetic variation and so forth. So from that period, it's now, I guess it's now 6 years since that first draft appeared and 4 years since the finished genome, we've continued to build the enterprise of being able to sequence DNA in the way that we learned to do it in the context of the Human Genome Project. So for those of you who've been upstairs, and if you haven't, you're certainly invited to come and look, we now have this big machine. It's 3 floors of this building, and it has 200 people, and it has, a floor full of instruments and computers, and its job right now is to produce about a 1 times coverage of a mammalian genome every month. So, in some contexts that's remarkable, because through the Genome Project we started doing a very tiny fraction of that and wondering how we could do this entire job. Now we can cover a whole human in 1 month. Normally, for reference, when we sequence a mammal, such as a human, we would do 6 times coverage. So for 6 months, in 6 months a facility like ours can actually push out a human genome sequence, equivalent to what the Human Genome Project offered, at least at its first phase. And so that's a number to keep in mind, please. We have a 3 billion base genome. You have 2 copies of that. You have a 6 billion base, 3 billion from your mother, 3 billion from your father, and that's about the number of raw bases we can push out from the Genome Centre. Now, since the finishing of the human project, we've actually spent more time sequencing other genomes in other creatures in order to inform what the function of the human genes are than we have put our energy directly into sequencing, again, different humans, for example, in disease context. And this is a small part of the menu of all creatures that have been pushed into this pipeline, and each one has its own story and own justification. And together, I think, with the other centres and other parts of the international program, there's probably 100 species, larger genomes and far up the tree that are well advanced in having their genome sequenced, and of course many more hundreds if you count microbes and small eukaryotes. So today I'm barely going to talk about any of these, but I do like to just touch on a couple of them to give you this flavour of how we've still got, on the genomic side of this genome medicine equation, we've still got a genomic enterprise that is about sequencing the genomes of animals to inform us through biomedical research and through comparative genomics. Example number 1, this creature here, the Tamar wallaby, which my friend and colleague Malin Renfrey here is catching, and there's its embryo. And I told a remarkable story, I think, at our meeting last week, how all these are born on the same day of the year, all the members of the species. So that's one piece of remarkable biology. But the other thing it didn't talk about is the antibiotic requirements for a creature that essentially does its development in the pouch rather than in utero. These are not placental mammals. These are marsupials. So this thing develops most of its early life period outside and or in the pouch. So at that time, it has a brother or sister who shares the pouch. So it has 2 teats, one which produces a remarkable range of beginning to be discovered antibiotics that can allow it to survive in that period, and the other which has other nutritional requirements. So what an opportunity for investigation in reproductive and early life biology. Here's another one of these creatures. This is the sea urchin, which probably some of you, or a few of you, might have worked on in some lab rotation at some stage. Of course, it's a model for development. It's another not too far distant eukaryote. And amongst the many things that we discovered in this long drawn-out genome project was that there were 80 or so genes in the creature which really can now be used and studied as disease orthologs. If you're working on a particular human genetic disease, you know the gene, you can now go to the sea urchin to be one of your tools in your set of creatures that you want to look at in order to discover the biology of that gene. And the last example is the macaque, which we finished recently, as David mentioned. These, of course, are extremely popular and important laboratory models, and they're used in neuroscience, they're used in vaccine studies, and we're going to have here we go. But they're also used in behavioural studies. What fabulous creatures and how similar to us they are. So for those of you who own dogs who probably think that dogs are almost the perfect models for human behaviour, it takes very little for a non-primatologist to see these primates in action and start to engage them as fellow creatures in their kingdom, for which we would love to understand much of their dynamics and how they work. So we studied the macaque and along the way deciphered just through its raw genome sequence and placing that in the tree and comparing its sequence to the chimp and to us, features about the rate of evolution of the different chromosomal events. It turns out, as chromosomes have changed across the tree, the changes have slowed down a little bit in this this part of the lineage here, close to us, like that. Right, more so than in the other primates, and that with other information is very informative about the dynamics of the change of these genomes across evolution. This is a long study, I'm not going to tell you much about it. Another highlight was the few genes were able to eke out of that genome by comparing it to chimps to humans and asking, of the changes that we see, how many of those changes reflect the pattern of action of positive selection across the lineage? Because those are the genes, of course, which we suspect are most important in saying why we're a chimp, we're humans, and the chimps are chimps, and macaques are macaques. And so this is part of that short list, and some of those genes have even been duplicated as the genome has gone through its efforts to make that part of the genome more conserved and more important through the evolutionary process. So we're continuing, of course, all these studies in these other creatures in order to drive our use of sequence and genome analysis to really basically inform human biology, to make more sense of the human genes, to make more sense of the human phenotype, and to allow us to make more inference. When you talk about something to do with the human gene, what does it mean? What does it mean for the human biology? So in saying all that, I kind of, and as we're all geared to do in the genome world, is to kind of make it sound easy. Okay? But in fact it's not, and it's still an expensive and clumsy growing science to produce whole genomes. And without going into the ugly details to prove that, or to really exemplify it, I'll just tell you that it still costs, to use those machines upstairs, it still costs about $10 million to generate that raw data. Right? And it still takes all those 6 months, which is a remarkable thing since it was a 15-year project through multiple centres the first time, but it's still 6 months, and that's no diagnostic test, right? And that's just to generate the raw data. You've still got to get a team of people that you can barely find and train to do the assemblies and put this stuff together and do the quality control and then annotate the data and then do all that stuff that makes a genome maybe the level of the macaque genome in terms of its utility. And if you want a genome like the human, which is actually a very refined and high-quality piece of scientific product, it's about the same effort again to get to that finished state. Okay? So the bottom line here is this stuff is still a raw and underdeveloped science. There's still a lot of work to do just in this basic effort of genomics. Having said that, there are still the programs have not been restrained in what we and they are trying to do with sequence as a basic discovery tool. And that's illustrated in a couple of things I'm going to just mention, one slide each, but not talk about in any detail today. For example, the copy number variation studies, Jim Lupsky is going to talk in this series, and what he will tell you is that following up his earliest discoveries and demonstrations that genome duplications indeed could drive genetic disease. It's quite controversial. Jim really nailed that maybe 10 years ago. That plus the observation now by many others that indeed parts of mammalian genomes are naturally polymorphic in their quantity. That is, you can look along the genome and find a part which is duplicated in one person maybe 3 times, and you can go to another individual and it'll be duplicated 5 times. And these can be large pieces of genome with function, with genes contained within and so forth. So that concept of copy number variation, now CNV, plus this notion of the engagement of disease has led to other programs which in some instances are using microarrays and other techniques, but in other instances are actually using sequence to drive the discovery of a full catalogue of those events. That's one program now going on. Here's another one. We actually have a lot of this anchored here through George Weinstock's efforts, and he's going to talk to you too, called the Human Microbiome Project. And this is the idea that since sequencing of bacteria is getting pretty routine, let's sequence them all. Now, of course, you can't culture many of them, so that provides a big problem. And so the initial thrust is to sequence the 600 or so that are in the catalogues of all culturable microbes, bacteria, and use this reference sequence plus additional samplings of different body sites, different orifices and different groups of people, and doing raw sequence, obtaining raw sequence to better understand the ecology of these positions in the body. So this catalogue is being built, and this is now an NIH roadmap program. This is going to be a big deal, and this is going to impact somehow on this whole concept of genomic medicine when we move past the human and the human genotype into other areas of sequence and those kind of activities. And of course, here's kind of the mother of all additional projects, the Cancer Genome Sequencing Project. This is Another NIH initiative that was launched a couple of years ago, which was the idea of let's extend this sequencing model and let's sequence a whole bunch of tumours. Let's extend our notion of characterising different cancer diseases to a full and comprehensive understanding of genomic changes and the range of genomic changes that can affect those diseases, and let's use that to drive discovery of what loci might be important in those diseases when we haven't found those loci already. And this is controversial and well worth at least a 1 hour group discussion and meeting already, but suffice to say it's off the ground. We have trials. There are 3 diseases that have been chosen by the larger program, glioblastoma, ovarian and squamous lung carcinoma, and the GBM component is going along quite well. There's actually another whole component that was funded by a pilot before this, that's lung adenocarcinoma. So this is all really rolling along. Here actually, in addition to participating in those large multi-centre programs, we have an initiative with the Department of Surgery where we're looking at pancreatic adenocarcinoma and looking at both sampling some of these candidate genes to look for mutational activity in them, but also really just generating the pipeline where we can reliably and meaningfully move samples from the surgical suite to the lab to get these initial diagnostic profiles. So we're doing what I'd call a model of how one day we might have a true diagnostic pipeline that is meaningful, where you can use biopsy instead of large tumour sections, where you can use DNA amplification techniques that are novel and have robust and meaningful results. So this is both a pre-test of that model as well as trying to inch pancreatic adenocarcinoma, and the group here, of course, closer to being one of those 50 diseases at the top of the list for the national program. So that's the applications in some of these big arenas of using sequencing to drive the knowledge. But what about human genetic disease? Because that's, remember, how we started here and what we're really focusing on today. Well, it's most useful to really think about human genetic disease in kind of 3 categories. And those categories are rare disease and rare disease, rare alleles, common disease, common alleles, and the stuff in between. Now, all of you I know are familiar with OMIM, with the catalogue of Mendelian disorders and Victor McCusack's list of, there's something like 20,000 entries, which boil down to something less than 10,000 opportunities to find human genes where you might find a mutation that can link that mutation into a phenotype. And about 2,000 of those are actually solved now. Well, all those disorders, or the vast majority of them, on this curve belong over here. Okay, so this curve is the fraction of SNPs, say, not the number, the fraction here of variation. I'm supposed to be green thing. And down the bottom is the population frequency. So a rare mutation, one that is private to your family or your near family, would appear on the left-hand side of this column, this graph. And that's where all the Mendelian disorders are found for the most part. Now, in general, think about those for a diagnostic challenge, okay? If you've got a Mendelian disorder that a family reports because of a cousin had the disease and you have no one else there, you've pretty much got to do the full gene ascertainment in general for most of these disorders. Okay? Now, a critical question in thinking about developing those diagnostics further is whether or not that will always be true. Okay? That is, will it always be a different mutation in those groups, or will sometimes there be a recurrence of certain ones? And so John Belmont with us has tackled this question because of what I just said, and also because we really don't know that much about these rare functional alleles and how they're distributed in the population, because we only ever see them when a family comes in with a disorder, so you get this real bias of ascertainment of what the true frequency is. And so John's done 2 things, and he's going to report these to you in detail, but I'm going to give you just a very high view of it. The first one is that if you look statistically across the larger databases at all these disorders, you see, although there is a preponderance of unique mutations in the databases, there are certain ones that do occur more than once. And since we have this bias of ascertainment, you can infer from that that maybe those sites are either present in the population at a very low frequency for some natural reason, something to do with selection or population drift, or they're actually mutation-prone sites because of the sequence context. So if you had a technology that was very efficient in screening for known mutations, you might want to include some of those in your repertoire of mutations that you screen. And this is the frequency in certain disorders. And here's the test that we used in a large genotyping project that John adapted for this exact purpose. He took 1,232 distinct mutations from a mutation database that were in 119 disease genes, which currently are screened for in the genetics diagnostics clinic. But they're screened for mostly by sequencing. Mostly they just look for the de novo mutations. And he went and made probes for those and went and asked directly were they in different populations. And the story he's going to tell you is he found some very interesting patterns that a subset of them at least were actually more frequent in different populations, and many of them were just overall turned up more than you would think. So this is actually pretty profound because it alters the way that you might want to generally approach screening. If you thought you always had to sequence to get a certain class of mutations, it's true. It turns out it's actually some of them at least you can put in an assay that is predictive for that particular mutation. That has tremendous implications in terms of implementation of this kind of work. But so much for that side of the curve and these rare mutations. What about the stuff on the other side, the common disease, common variant mutations? Now, these are the ones I think that are driving all the excitement in this whole notion of genomic medicine, or at least genetic genomic medicine in the first place, because there's the idea that you can get a predictor for, going at the age of 20, get a predictor saying that you're going to have a heart attack when you're 50, so you better do something different than if you didn't have the predictor. Okay. Now, faith in that depends on this whole notion that there is a thing called a common disease, common variant hypothesis. What I just said is not quite true, but practically true, and you'll see why I qualify that in a moment. That it's the idea that there are actually alleles out there that are fairly common in the population, which if you happen to get a bad combination of them, they give you this propensity to have one of these disorders. And those alleles, by their very nature, have to be common, otherwise the whole statistical model wouldn't fall together correctly. Now, another follow-up to the Genome project was the HapMap project, and this project was designed essentially to allow us to test that hypothesis and find out if there were indeed alleles out there that you could assign as predictive for these kinds of disorders. And the idea of HapMap was to take natural populations, reference populations, and find all the variation you could given the available technology, and then map that variation across the whole population, and then use it to tag different parts of the genome. So the underlying idea here is if humans aren't that old, we've only been out of Africa for 100,000 years or something, 10,000 generations, maybe less, and so our chromosomes haven't had that much time to reassort. And if you pick two ancestral chromosomes, even through that many generations, in the current populations, you can see large blocks that have been conserved over that whole period. And this kind of genetic discovery and mapping can allow you to track those blocks in an efficient way. So that was HapMap and all the hype of HapMap. And then, so the big experiment is this. Sequence the genome, do the HapMap to find all these referent variants, and then take those variants and genotype them in critical populations. In your cohorts of diabetics versus matched controls, your cohorts of schizophrenics versus matched controls, your cohorts of Crohn's disease versus matched controls. So now that experiment's been done. We've been waiting with bated breath for this for at least as long as the Genome project has been going. The Wellcome Trust funded the biggest study and the one that's had the most publicity and the most success. And many of you might have seen this figure in Nature a month ago, and we've all been going to meetings and celebrating tremendously. This is what happens if you do that experiment. And you look for parts of the genome in these populations that have an unusual genetic conservation signal that reflects some increased, some propensity of that part of the genome to drive an increased risk for one of these diseases. And you see those little green bars there in the different disorders. Those are the success stories. Those are where there is actually signals from these disorders. Now, once the champagne was finished and everybody sits back to think about it, it is striking and it's stunning and it's a fabulous result. But there's a lot of qualifiers here. So you might ask, as a clinician, does that mean you're ready to run and use these markers and counsel people based upon these markers? Well, the odds ratios, that is the propensity of you to have the prediction versus not, if you have the allele or not, range from about 1.1 to 1.6. Okay? So your frequency, your likelihood of having a heart attack might go from 1 in 3,000 to 1.6 in 3,000 from these markers. They're really not that much use when you distill things down to that level of activity. What they are, of course, incredibly useful for is saying there must be a gene under here. That gene must be involved in a pathway. Other mutations in that gene might be more profound. They may be driving the disease. So this whole approach, looking for common disease, common disorders, enjoying very good success right now. but it's still a tough game. Here's one zeroing in on one region. This is one of the most exciting stories in the genome right now. One of those regions that was tagged was on chromosome 9, and it's 50 to 100 kilobases, and it came up strikingly in the diabetes cohort and in the cardiac disease cohort. Okay, and it turns out there's only 2 genes in the regions: there's CDKN2A and CDKN2B, and they're well known for having mutations, somatic mutations in pancreatic adenocarcinoma and some other cancers. They're the only genes in the region. So this has got everybody baffled. What's really going on here? So the dream, or the hope is that either another gene, or maybe a gene controlling element, or some other part of the genomic biology here will emerge and explain all these phenotypes, and will have larger effects when there are different changes that are found in those same elements. But right now, this is the state of the study. Okay, so what about the rest of the whole spectrum? And this is where the things get kind of interesting, because this actually turns out to be where most of the action is. Although there's been celebration of the success of the HapMap and what I just told you, it's also abundantly clear that an awful lot of the variation is falling into this middle category. It's lower in frequency. As an individual event, it probably has more effect on phenotype, and it may be therefore more interesting, say in the clinical context, but it's harder to find. Now, there was a lot of excitement was generated about this class by our colleagues at Southwestern, Helen Hobbs and Jonathan Cohen, who looked at various lipidemias and are looking at various blood lipid levels, and in their Dallas Heart Study cohort, separated individuals out according to that distribution that you see, and just focused on candidate genes in pathways at the 2 ends of the distribution. And they actually showed, I think, maybe not for the first time, but most dramatically, that there were certain genes that simply had a lot of different mutations. Low frequency, 1% in the population, 0.1% in the population, had a lot of those at one end of the distribution, but not in the other. So if you're willing to go out there and sequence a lot of these genes in people at the 2 ends of the distribution, you could start to bin them according to genes that had a higher overall mutational burden than others. And because they have the function and the biology to back this up, the story really makes sense. So many of us have been setting out now of the other studies we're looking at to try and fit them into this paradigm, okay? Because we know we're not dealing in these other studies with other diseases with common disorders, and we're not dealing with Mendelian forms. We're dealing with something in between. And here we've worked with Jeff Nobles on epilepsy, and this just illustrates the range of possible causes, including some unknown genetic cause. It's the biggest piece of the pie there. We've used the sequencing methods, sequenced about 200 people in about 240 genes that are in the candidate set of ion channels, and we've found nearly 1,000 changes in those people that might cause the disease. So what we're doing now, I said might, not does, we're trying to figure out amongst those changes, are they distributed statistically, is there functional clues, these are the knockout mutations that kill the genes, or the other functional clues about the number of individual mutations in those loci in the cases versus the number of individual mutations in those loci in the controls. We're trying to distill out from that whether or not we can really assign any of these genes as being truly part of the epilepsy syndrome that we're chasing. I want you to get the flavour of how difficult all this is. We're occurring heterogeneous mutations in diseases with incomplete penetrance, with really uncertain genetic models, and trying to assign these mutations now at the quality of a diagnostic marker. That's a big gap, right? So we have a few stories like that now, all these tough unsolved genetic ones that we have a lot more genetic variation data. Now, there's some good news here, we think, which really comes from the technology evolving rather than particular instances of solution of the genetic paradigm. And they come from, they are to do with the notion that maybe we don't have to be too obsessed about solving these problems one at a time. Maybe we can actually go back to these reference populations and sequence all the exons in everybody. And then maybe, even in 2 years, we're talking maybe we can sequence all the genomes in everybody and have this several thousand genomes deep database that will really give us a map of these rare variants and make these kind of questions about what's really segregating with these diseases with very complex genetics more straightforward. Okay, so that's our and there's even more good news, and I don't want to get into technical nuances, but we've been slogging away at this with PCR and with Sanger sequencing, just like I showed you in that slide from 1989, which is like a long time ago. And only in the last 6 months do we have a leap in the technology we think will change things, and it's the idea we can use microarrays with targeted capture oligonucleotides on them that will pull out the genes of interest and allow them to take these things off the array and put them into the new generation of sequencing technologies and give us very rapid determination of the small part of the genome we think is the target in these groups. So that's the good news. We think we can build this big catalogue. Okay, so that's 1, 2, 3. That's the rare alleles, that's the common alleles, and it's how we're tackling this group in between that's so important. But the thing about allele frequencies is that when it comes down to individuals, or you or your patient, the frequencies are really either 0, 1 or 0.5, right? You're really not talking about a population question. You really have to do some kind of ascertainment that gives you very high quality data about individual samples. So within all this muddle of different activities, we have been also steadfastly focusing on this notion as when can we deliver this idea that an individual could come in and get a meaningful genome of their own. The snapshot of the cost of that today is probably, this slide's out of date, it's probably only $15 million there to do it the whole genome way. But to do all the exons is about $400,000. Okay, if we were to, you were to get all your 300,000 exons, and we're missing a lot of the stuff people always says is important, the intragenic stuff. 300,000 exons to do PCR and to do that Sanger experiment, about $400,000, and probably could do it in about a month upstairs. So that's clearly too expensive. So the big question we've been facing is where are these new technologies going to fit and how will they enable that kind of effort? And here's the current repertoire of the new technologies, and actually 3 now, which are having some impact. The Agincourt AB one at the bottom is probably going to be a major player in the near future, but the 2 we have experience with now are called Celexa or 454 Corporation. And the Celexa method is, its ideas have been around for a long time, but they've just been enabled by clever chemistry and clever enzymes. This is the idea that instead of doing a Sanger reaction and using a gel or a capillary, what you do is you have a very small flat surface and you spot DNA on that surface in a very dilute fashion, and then by a surface-linked polymerase chain reaction, each of those molecules is amplified and grows to form a spot around the point it landed on. So then you have a sequenceable molecule spaced through this little chip. Then you come along with a set of 4 nucleotides that, through very clever chemistry, both terminate sequence reactions, report their own fluorescent colour, and then by other treatment can be made to bleach out the colour and then let the reaction proceed. So what you end up with is this big field of molecules that you can treat with all the 4 bases and will be turned a particular colour depending on what the next DNA sequence base is. You bleach it and wash it, do the process again, the next base is reported in each molecule. It's quite amazing technology. Now, what's the problems? It takes 3 or 4 days to do a run on these machines. And the error rate is really quite high right now, and there are a lot of just general practical problems in implementing it. Chemistry, enzymology is very difficult. But this gives you an idea of where things are going. This machine, when it works properly, could produce 1 billion bases in a run for about $3,000. Now, the other option that's really out there is the 454 machine, which uses a slightly different approach. Individual DNA molecules are captured on a bead, a very small bead, and then amplified around that bead in a test tube that's filled with oil and water, and little aqueous bubbles covered in oil form a little reaction chamber and allow that one molecule to amplify around that one bead. So you take your DNA, shear it, put it in the tube, do that, and then extract the beads and then put them into a flat chip that is actually 1.3 million wells with little etchings on it. It's glass fibres, fused, shaved, polished, etched. It's quite extraordinary. So those big beads in the bottom there are actually the beads with the DNA on it. The little beads are just packing beads. So that thing is pushed up against a camera and the chemistry is done and you get to produce the DNA sequence. And you get maybe 100 million bases per run. Those old machines from Applied Biosystems, 100,000 bases. So you see the growth in the technology. Now, those are the 454 machines in the foreground and the Celexa machine in the background. And you can see they're upstairs if you want to come and see them. And it works pretty well. The 454 we have a lot of data on. These graphs just show you that the error rates for the machines are down on the low end. And depending on the particular genome that's used, can give you a product that's used what we're used to getting from the Sanger reaction. So this is what we used in the Watson project. Now, specifically, our collaborators at 454 did the actual sequencing and paid for it, and went and did the Watson project as a stunt project to get some publicity, which sure worked. You've got to, whatever else you say about the project, that's certainly successful. And they generated all the data and allowed us to do the analysis. Now, Amy, who's in the audience, who's going to talk in this group a little later, helped us manage the ethical interactions with, the data release concepts with the company. One of the premises of the project was that the participant would release their data would be inclined to release their data. I've got to choose my words very carefully. That wasn't an absolute requirement. Somebody who would be inclined to release their data to the public arena, just as we've been doing in the Genome project all along. And Dr. Watson certainly was willing to do that. And through the course of the project, a lot of discussions emerged about really what that meant, and that it might not be as benign of a thing to do as you might think. So let me do the quick quiz. Who here would put their data out, their complete DNA sequence, on the internet without any qualms or thoughts tomorrow if we offered to sequence you in the Genome Centre, if you would do that? Are there any takers at all? 2, 3. Okay, let's sign you up. So the risks, of course, aren't fully. The model is that the risks aren't fully known. You may be incurring some problem for your family. Of course, remember you share your DNA with your family, and you might be showing their propensity to do certain things that you might be completely proud of, but they might regard as, they might regard as something they want less out there. So in any case, it's a complicated question. And so what we were able to do, and Amy designed this data flow, was that we managed to make it clear that the submission to the public databases would be from the individual who had his sequence given, his sequence obtained. So here we were able to do the analysis and validation of the data, and it was passed back to him for him to release to the public domain. Actually, someone sent me a link. You can actually download it right now if you want from the NCBI. It's very accessible. Now, we did an analysis here in the genome, and this slide's just to remind me to tell you that 4 people did it in 3 weeks, as opposed to the macaque genome analysis, where 180 people actually did it over a course of a year. Right? So the analysis we got from Watson was less comprehensive in many ways than what we're able to do with the other major genome projects. Nevertheless, the fact we were just aligning him to a reference sequence made many of the questions easier. But here's the very high points of what we found. We found a lot of individual base variants. We've actually redone these alignments, and the number's a little higher than 3.4 million in the complete data set. So about the expected number of overall single base variants. Amongst those, though, there were about 6,000 that clearly have the potential to alter the function of those genes. And those of you who think about mutation frequencies and mutational burden will actually may or may not think that's a high number. Amongst those, there were 300 or so that clearly were in genes that were in that disease list. And then 23 of them were actual alleles that were known, that had been reported before as bad alleles. Right? So what was really exciting about the project, here's some of the genes, and I bet if I leave that up, some of you will find genes that you already know quite well and will wonder what the mutations were in this individual. So a really positive thing about the project was the taxing nature of it for the genetic counsellors here, who are the best in the world, frankly, at single locus diagnostics. If you take a family with a disorder that someone in the genetics group here is familiar with and tell them about the mutational spectra, there is no better people to go to talk about those mutations. But if you give them a data set or a spreadsheet with a minimum of 300 genes and a maximum of maybe 6,400, where there are potential problems, that's a very different paradigm for them to deal with. But in fact, it's the one that we're leaning towards and moving towards as this work emerges. So here's our summary of the Watson project that, it really has pressed the model, the concept of a genetic test and just how many mutations you can find if you look across the whole genome, and how indeed you simply handle that data and how you show it to the individual and how you show it, share it with each other. And so a good demonstration project. So I'm nearing to the end of my presentation here. So I wanted to bring it back, though. If you reflect on all of that, what should we do here then to catch this wave that is coming or crashing, or depending on your perspective and how mature you think the data set is? And one of the most interesting sets of discussions that's been going on in the college for at least a couple of years has been the different efforts to collect samples in a more systematic way to make them more available to individuals. And several of you in the room have been either involved in those discussions or you have your own collections, and you have talked with others about how you might share them and how you might bank them. And I wanted to encourage you to go and look at the University of Tokyo and Biobank Japan and Professor Yusuke Nakamura here, who, as part of the HapMap project, began biobanking in their clinic. And he has now, I think more than this. I just pulled that number down last night, 115,000 samples from visitors, all with good phenotypic records, and all with, in their case, they've done multiple blood draws. So they have it's 115,000 individuals and multiple draws on the same individual. So they have DNA and they have multiple samplings over time. I don't know if they have individual tissues. But I think these are a set of questions that we want to raise here for consideration and for maybe thinking that we should be collectively encouraging the college to put resources into. I saw him present at the Hugo meeting in Canada last year, and he talked about warfarin sensitivity, and of course I'm probably the least qualified in the room to talk about this, but I discussed it briefly at a meeting last week, and I said I understood it was an empirical science to approximate or to get correct the dosage with which you would give this class of drugs to particular patients, and the response was, no, it's not a science, it's not even empirical. Okay, so I guess it's tough, right? And the point is now there are certainly known genetic markers that influence, if not direct response, long-term response to these drugs, and the long-term coagulative response, and there's good literature on that. The point is, I guess, that in Japan they are certainly typing these individuals for those markers ahead of time, so that as part of their clinical record they can have that anticipation of what this might influence their therapy. What a good idea. So the other thing here, we've had some good discussions, and hope some of you met Tony Elam. He's helped us a lot here too, about expanding our overall and coordinating our overall data farming usage. There's no question, and the Watson project brought this home more than anything, that data crunching, data handling is still a wall that we're always pressing up against. And I think as part of our future and as we move into this new clinic, it makes perfect sense that we should have some kind of log leap between now and then in our capability of doing that, just to give us the elbow room to do what we need to do as these kind of data start to flow on. So here's something of the model, how this might fit together. And I'm sure many of you could add to the model. That is the idea that if you were facilitated in your clinical and your clinical science practices for sample archiving, and doing that in a way that was commensurate both with the patient record requirements and with the ELSI requirements, then that would be a great asset both to yourselves and to the research community here, who presumably then could, through a proper but minimal protocol development of protocols, access those sample collections in collaboration, but not without the onerous mechanics that are currently needed, access those samples and the accompanying data they were allowed to get in order to do research activities that were not moving mountains, they were simply accessing this large data body. I think that sounds like a pipe dream, but that to me is almost a prerequisite for pushing us into this next phase. And of course the notion that genetic diagnostics, I call it plug-and-play diagnostics, those assays that are already calibrated and working that you can access, of course, will be an intimate part of this, but really we want to press that up against these developing techniques. I mean, you can't get a genome sequence now, but the minute you can, you want to have it somehow running through into that plug-and-play arena there, so it can be properly used by the clinician. So I think these two elements are really things that we might want to all press on. So in grand summary then, I would conclude that things are moving pretty quickly. I don't think that we have that many home runs to really point to in terms of alleles that were not known yesterday, but today everybody wants to have them screened. But I think this groundswell is truly happening, and I think we can just point to the databases and the literature to say that that's true, and we should brace ourselves. Now, thank you everybody, and of course there's many people here I hope that you know or might get a chance to meet who worked on these different areas. And this is the Genome Centre staff, and thanks a lot for listening.